The materials used in this experiment is paper, 2 spoonfuls of milk, cheesecloth, 30 ml of warm water, ethanol alcohol, rubber band, and vinegar. Measure 5 grams of whole milk or 2 spoonfuls in a clean container. Pour 30 ml of warm water into the milk and mix. Next, add drop by drop acetic acid, vinegar, while stirring the milk suspension. Observe the formation of precipitate. The purpose of adding vinegar is to lower the pH of the milk for the protein casein to reach its isoelectric pH. When that occurs, the casein will separate from the solution. Filter the mixture into a cup by pouring it through a cheesecloth, which is fastened with a rubber band over the mouth of the glass. Remove most of the water from the precipitate by squeezing the cloth gently. Discard the filtrate. Scrape the precipitate from the cheesecloth into a clean container. Then, add 10 ml of ethanol. Keep stirring the mixture for 5 minutes. Because ethanol is less polar than water, it lowers the electrostatic interaction that help play a role in stabilizing the casein emission. Filter again using the cheesecloth. Scrape the casein on a paper. Spread the casein on a paper and let it dry. So this is the result of our isolation of casein. It is very thick and it kind of reminds me of a lumpia wrapper. So here is our experiment.